Hello everyone. So today we will learn about culture media, the one that is used to grow different kinds of bacteria. So it is basically divided into four types, that is simple or basal media, enriched media, selective media and differential media. So simple or basal media is only used to grow non-fastidious organisms. Example is nutrient agar. So another enriched medium. So enriched medium, it is enriched with nutrients to help the bacteria grow better. So it can be nutrient agar plus egg, blood or serum. Example is blood agar. Selective media, which means that it prevents the growth of unwanted bacteria. Example TCBS, clade, McConkey, mannitol salt agar, etc. And differential media is used to distinguish between bacteria based on biochemical reactions. Example McConkey agar. Other are anaerobic media, which is used to grow anaerobic organisms, and transport media, which is used for transport of organisms. So let's start with simple or basal media. It only allows the growth of non-fastidious organisms. So examples are peptone water and salt, NaCl 0.5%. So peptone water can be 1%. Now if we add 1% meat extract, it will become nutrient broth. Now to this nutrient broth, if we add 2% agar, it will be nutrient agar. Now let's move on to enriched media. First one, that looks red. So what is red color? Blood. So it's blood agar. If to nutrient agar we add 5 to 10% sheep RBC and autoclave it at 50 degrees Celsius, we get blood agar. And if we autoclave it at 70 degrees Celsius, we get chocolate agar. Simple. It's used to distinguish between hemolytic and non-hemolytic bacteria. It contains 5 to 10% sterile sheep RBCs. And it is autoclaved at 50 degrees Celsius. Thus, blood agar is formed. Just to be clear, the 5 to 10% sterile sheep RBCs, they are added to nutrient agar to make it blood agar. The next one, that is chocolate agar. When you see a brown agar, think of chocolate, and that's the name, chocolate agar. So the one that is autoclaved at 50 degrees Celsius is your blood agar, and the one that is autoclaved at 70 degrees Celsius is your chocolate agar. It is also made up of 5 to 10% sterile sheep RBCs. It is more nutritious and it releases factors like factor 10 and factor 5 for Haemophilus influenza. So let's quickly revise blood and chocolate agar. If to nutrient agar we add 5 to 10% sheep RBC and autoclave it at 50 degrees Celsius, we get blood agar. And if we autoclave it at 70 degrees Celsius, we get chocolate agar. Simple. And chocolate agar, it has an additional property, that is, it is more nutritious and it releases factor 10 and factor 5 for H influenza. The next one, that is Loeffler's serum slope. Only thing to remember about this is that it is used for Corynebacterium diphtheria and it gives result in 6 to 8 hours. And the last one, that is Lewinson Jensen media. So if you see a media that is macellite green in color with rough buff colonies which are creamy in color, that is used for Mycobacterium tuberculosis. It is enriched by coagulated hen's egg. Now let's move on to another type, that is selective media. It inhibits growth of other bacteria. The first one, that is McConkey agar. It is also a differential media because it can differentiate between lactose fermenting and non-lactose fermenting species of bacteria. If the colonies are pink in color, they are lactose fermenting, and if they are pale in color, they are non-lactose fermenting. The next one is TCBS. This is used for specially Vibrio, V. cholera. It produces yellow or yellow golden colonies. It contains bromothymol blue as an indicator. It is a differential media that can differentiate between sucrose fermenters versus non-sucrose fermenters. And most importantly, it contains bile salt that makes it selective. The next one, that is mannitol salt agar. It is a differential media that gives a yellow color on mannitol fermentation, and it is selective for S. aureus, and it has salt which only allows S. aureus to grow. Indicator used is phenol red. It is also a differential media as it can be used to differentiate between S. aureus and Staphylococcus epidermidis, which is a non-mannitol fermenter. The next one, that is CLED. Stands for cysteine lactose electrolyte deficient. The yellow colonies on the agar indicate lactose fermentation, and the indicator used is bromothymol blue. So it is also a selective media and a differential media. Now there are some other media that are specific for some organisms. So let's learn them. There is DCA, that is deoxycholate citrate agar, and XLD, that is xylose lysine deoxycholate. It is specifically used for Salmonella and Shigella, and it gives us pale colonies with black center and red colonies with black center. The second one, that is potassium tellurite agar. It is used for Corynebacterium diphtheriae. It gives small black or brown colonies. The next one, that is crystal violet blood agar. It is used for Streptococcus pyogenes. The next, that is modified Thea-Martin medium. It is used for Neisseria species both, that is Neisseria meningitidis and Neisseria gonorrhoeae. The contents of modified Thea-Martin medium were actually asked in NEET PG or INICT, so it's an important PYQ. So it contains vancomycin, colistin, nystatin, trimethoprim. Then cetrimide agar is used for Pseudomonas, and plet agar. Usually the full form is not asked, but it is polymyxin lysozyme EDTA thallose acetate. It is used for Bacillus anthracis. Now differential media. You can see there are pink colonies and pale colonies. It is McConkey agar. Pink colonies are for lactose fermenting, and pale colonies are for non-lactose fermenting bacteria. The next one, that is clade agar. It shows yellow colonies for lactose fermenting. This is blood agar. It is used for differentiation between hemolytic and non-hemolytic organisms. Example, the Streptococcaceae family, or the basically Streptococcus, on the basis of hemolysis on the blood agar, it is divided into alpha, beta and gamma. So alpha hemolytic is Viridin streptococci and Streptococcus pneumoniae. Beta hemolytic is further divided into 20 groups, but the more common are group A, that is S. pyogenes, and group B, that is S. galacticae, and gamma hemolytic, that is Enterococcus. Transport media used for specific organisms are: for Neisseria, we have Stewart's media and Amy's media, and these are charcoal based medium. So for Vibrio, we have VR medium and Carey Blair medium. For Streptococcus pyogene, we have only one, that is Spike's medium. And for Salmonella and Shigella, there is Sachs buffered glycerol saline. Now anaerobic media. Anaerobic media, the first name that should come to your mind is Robertson's cooked meat broth. So if you see any bottle, test tube, or anything containing a little bit of yellowish, some oily medium along with some pieces of meat in it, it is Robertson's cooked meat broth. It is mostly used for Clostridium. It can also differentiate between Clostridium tetani and Clostridium perfringens if the meat turns black. It is Clostridium tetani. It is proteolytic. And if the meat turns pink, it is Clostridium perfringens. It is saccharolytic. So I hope you have understood this well, and this video helped you in any way to understand the classification or the examples. I hope you enjoyed this video. If you did, please like, comment and share. Thank you.